Okay, without further ado, I want to introduce the first speaker and organizer and creator of the idea. He will also explain what the idea is with this picture on the screen. It's Thomas Heimburg, we have a lot of know each other since quite a while, because I was one of his first diploma students back then in Göttingen. I also met Konrad Kaufmann and Erwin Niels, so that was the first three people that influenced me a lot, and he kept communicating since then. Okay, Thomas, thanks a lot for. Thanks a lot for being here, and you have 35 minutes for your talk and 5 for the discussion.
Okay, thanks very much for all that. It's an invitation. This little figure here I found by accident. It's called Poem, and the idea is that might be a kind of conference mascot, and if somebody else picks up next time, then it can be present. I just like that.
I think we should use the microphone.
Yeah. Okay. Nerves are typically depicted like that. You have a soma, you have dendrites and axons in there, and the nerve pulses are in these drawings always very, very small, which... Yeah. Can you hear me better? Can you hear me better? Typically people believe that nerve pulses are something very, very small, and that's why microscopic models are so popular in biology, because obviously you need small things to explain small phenomena. And what most people record in nerves is a voltage change. This here is a nerve pulse from a lobster, a neuron, which lasts about 1 to 2 milliseconds. So now, if you calculate the length of a nerve pulse, you find that a motor neuron has a velocity of about 100 meters per second. The time scale is about 1 millisecond, and you calculate a length scale of 10 centimeters for a nerve pulse. So that means in conflict with these drawings here, the nerve pulse is in fact something very, very large. In most nerves, it is on the scale from millimeters to centimeters. It's definitely not a micrometer scale phenomena, and that cries for the application of more microscopic models. We will have a roundtable discussion later where we will probably talk about scales, I assume. Of course, if you have a large-scale phenomenon, you would like to model it in physics with macroscopic theories rather than with microscopic theories, which is the attempt that has been made in biology all the years. So what is also known about nerves is that they show other features than just voltage changes. For example, they have mechanical features. They contract and change their thickness. This year, there are old experiments by Tazaki and Iwasa on that. There is also an experiment from a group from Pennsylvania that has measured thickness changes in synapses. This is our experiment. This is a lobster nerve going from the brain to the first ganglion, and this is a nerve bundle. When you open it up, you see a single neuron, and one can put an AFM needle on that single neuron and measure the thickness change, and you get something like this. This here is the differential change in voltage, measured by 2 electrons next to each other, which measures effectively the first derivative of the nerve pulse. This here is when you integrate it, that's a nerve pulse, and that is a thickness change in the nerve. So there is a region in the nerve where the membrane just is a little bit thicker, and then in the rest of the neuron. This here are just 6 representative traces from different neurons. It's absolutely reproducible. You always get these thickness changes, and the exact magnitude of the pulse varies a little bit depending on how close you get the cantilever to the nerve. But typical thickness change is about 1 nanometer, and that is about the scale that Tazaki measured also in something like nearly 40 years ago. Sounds like something very small, but you should remember that the membrane of a nerve is only 5 nanometers thick. So it represents approximately 20% of the change of the thickness of the overall membrane. So what has also been recorded, unfortunately not by us, because I have a very hard time to get funding for that, but I would like to reproduce that experiment, is that there is heat release during the nerve pulse. There is a first phase where heat comes out. The direction is important. First heat comes out and then heat is reabsorbed. And if you integrate that curve, you get a profile like this, which basically tells you that during the nerve no heat is dissipated and the phenomenon is more or less adiabatic. This is also an old experiment. The first good quality experiment comes from Archibald Hill from the 1950s, but the heat measurements in nerves trace back to Helmholtz in the 1850s. And so it's a very, very old observation. This observation that the heat is conserved, or there's no heat dissipation in there, led Archibald Hill already in 1912 to assume that the nerve pulse might be a sound-related phenomenon, a reversible physical phenomenon. That is a very old idea. To tell you what that means is, if you consider an adiabatic phenomenon, which is typically described by a constant entropy, that means ds is equal to 0, and you can write it down as ds dt and ds dx for a spring. I demonstrate that for a spring, right? And from that, this here is the heat capacity divided by temperature. This here is the mechanical force divided by temperature. And from that, you can calculate under circumstances where the entropy is constant, that there is a temperature change in there which is related to the energy in the spring divided by the heat capacity. And actually, I walked around with the Niels altitude and told them the temperature of the spring should change temperature, and nobody believed that because they said thermodynamics and analytical mechanics are two completely different theories applied to different phenomena, and that has nothing to do with each other. And then I just measured it. This is a big spring. One can attach the thermocouple to it, attach the mass. One can calculate how much larger the energy is, so one can choose the appropriate spring. And what you see is a beautiful oscillation in temperature. In this experiment, it was on the scale of a few millikelvin a few millikelvin. And what we calculated was something more like 50 or so, but it's very difficult to get a close contact of a thermocouple with a spring without creating an electrical contact. So that means you have to insulate it electrically in order to not get any artifacts. So one can probably do that better than we have done that, but this is a proof of principle. You get the right order of magnitude of temperature oscillation. So the claim in my presentation is whenever you see something like that, a periodic change or a reversible change in temperature or in heat release, then this is an adiabatic phenomenon, which also is important for the talk that Wolfgang Anke is going to give, because that is also observed in the Belousov-Zhabotinsky reaction. So now let's go to describe the thermodynamics of such a pulse. First of all, most of you know that in membranes you have melting transitions. You go from an ordered to a disordered state. You have enthalpy uptake, change in enthalpy. You have entropy changes. You have a melting temperature. During these transitions, you get changes in enthalpy of about 35 kilojoules per mole, volume changes, pretty significant area changes, thickness changes of about minus 16%, which is of the order of the thickness change that we see in the nerve pulse, which is also important. But remarkably, you also get significant changes in capacitance. We have measured that. You can actually, when you go through a transition, you can double the capacitance. Because the membrane becomes thinner, it gets a larger area. So capacitance are by no means constant in biological membranes. So I already said that biological membrane, not many people know that biological membranes also show transitions like that. This is a transition in lung surfactant. This here is one in the spine of pigs. We have measured that for sheep, chicken, rat. We have found these transitions in E. coli, Bacillus subtilis, and in all kinds of cancer cells. And so these transitions are practically always there. They're nearly always at the same position, something like approximately 10 to 15 degrees below physiological temperature. That seems to be something conserved. in nature, and for some funny reason nobody makes use of this remarkable fact that there is a conserved physical behavior in these membranes. So one can, when you know the heat capacity of such a pulse, you can also calculate the sound velocity. I showed you briefly how to do that. The compressibility has been shown to be proportional to the heat capacity, and for the sound velocity is described by that sound velocity squared is 1 over the compressibility times the density, and you have here the adiabatic compressibility. That means the compressibility that you have when you don't allow heat transfer to the outside. And that is described by this equation that contains the isothermal compressibility, the volume expansion coefficient, all kinds of functions that you can calculate when you know the heat capacity and you know the time scale of the phenomenon you're looking at. When you make a calculation from a heat capacity, you get a heat capacity profile that looks like this solid line. A sound velocity profile as a function of temperature. And at the point where the transition is, you have a minimum in the sound velocity. The dotted symbols here, the symbols, they represent a measurement done by a group in Göttingen, by Ude Katzer and collaborators, and which basically confirms that you can calculate sound velocities from when you know the heat capacities of the system. Now, you can apply that to two-dimensional systems as well. You can take the heat capacity, you can calculate from that the change in density, because the density is related to the enthalpy and the area of the system. The membrane increases it by about 24%, and that's reflected in the area density of the membrane. So it goes down in the transition, the compressibility shows the maximum, and you can in principle now plot the sound velocity, which is 1 over this quantity times the density, versus the density, and you get a profile that looks like this. On the left-hand side, the density here corresponds to a liquid phase. On the right-hand side, it corresponds to a gel phase. Yeah? That means if you go through that transition, you change the density, you get a minimum in the transition, which is then the main property of the mechanical phenomena that I'm going to show you in a moment. That is because of a discussion I had with Matthias. Matthias somehow said that we use a very weird deviation from the wave equation. I want to show you that this is not true. The wave equation in hydrodynamics, that is basically the equation that governs sound-related phenomena, looks like that. You have a second derivative of density with respect to time. And you have a diversion c squared gradient delta rho term in there. That's in 3 dimensions, right? And if you can, with that, you can describe, for example, what happens when you throw a stone in water and things like that, waves that travel away from the center. In 1 dimension, it is like that. Yeah? And what is important is that the sound velocity here is inside of that derivative. That is an important feature. If, however, the sound velocity is something constant, that's what we, for example, assume when we talk about sound propagation in air, yeah, then you can move it out of the derivative, and then you get the equation that we always use when we describe the propagation of sound. Yeah, so this is something first-year physics education. So now, there's another feature in there, which I described earlier here. At high frequency, actually this profile looks a little bit different. It is not quite as pronounced, and so that is because membranes become stiffer when you do things fast. Yeah, therefore, the sound velocity things are not quite as pronounced. And this effect, frequency dependence of sound velocity, that's called dispersion. Yeah, when you do things fast, things are slightly different, and membranes are stiffer, and sound velocity is higher on average. Yeah, so if we go here, one can do, in principle, take the sound velocity and say, okay, let's assume we can expand that in a Taylor series. It's a constant plus a linear term in the frequency plus a quadratic term in the frequency, and so on. And since you know for sine wave propagation, minus frequency and plus frequency should give you the same result. That means the linear term should cancel. And you have, as a first order approximation, a quadratic function in the frequency. Right? So now we take the wave equation here, in the case of when the sound velocity is not density dependent, yeah, and we add a term in there. We do that ad hoc. We just try it. Right? And what that is doing is, if you add a sine wave solution for the density, that you get an equation like that. So you can plug that in, and then you get k's and omegas in front, and you can derive this here, that omega squared divided by k squared is equal to C0 squared plus a constant times omega squared. Yeah? So that means if you add that term into the wave equation, then you actually reproduce the most simple dispersion relation that is possible, and that is what people do in the field generally if they want to include dispersion in a wave equation. And this is a trick that is generally known, and many people have used that in the past, so I didn't make that up. So the reason why we modified the wave equation here is to include dispersion. Now, I've told you already the sound velocity is also density dependent. That have what I've ignored so far, right? So you see that if you can in principle also expand that in a Taylor series. You say it's a constant plus a linear term plus a quadratic term and so on. You can use as many terms as you wish. So if you just stop with a quadratic term, you get fits like that to the profiles. Not very nice, but good enough for demonstration reasons. And then if we take this wave equation here that we have derived before, and we plug that Taylor series in, we get an equation like that, which turns out to have solitary solutions. That means pulses. Yeah? And this equation down here is the same equation, we just have transferred it in a moving frame. So that means we have a coordinate system that is always centered at the peak of the pulse. Yes, that's the only difference between these two equations. Now, the solitary solution looks like this, and what you have to consider is that we start at a density that is in the liquid phase, and then we excite the nerve and make it more dense. And the more we excite it, the more it goes through the transition, and we get a pulse like that, where basically the membrane is solid. Yeah, and interestingly, it has a velocity around 100 meters per second, which is at least close to the velocity of myelinated nerves. It has the feature that since the membrane here is solid, it will get about 1 nanometer thicker. Yeah, because that is the difference in thickness between liquid and fluid. So that's the order of magnitude of the pulse that we see mechanically. Right? This here on the right-hand side is just for comparison the lobster neuron case. That's an experimental thing. Okay, now you can also from the hydrodynamic theory calculate the energy. The energy of the pulse is like that. Yeah? And actually what I forgot to say is in this equation, which I showed on the previous slide already, there's a velocity in there. That's the velocity of the pulse. If you change that velocity, you can go from very small pulses to larger pulses, and then they hit a maximum, and then they get broader. Yeah? So if you have a very small pulse, that basically means that the system is still liquid. Mostly you don't go really into the transition, and you're more excited, the more it goes into transition until it goes completely through the transition, and then you don't get a further increase in density. You can calculate for each of these pulses, you can calculate the energy, and then plot the amplitude versus the energy, and then you get this kind of profile, yeah, which basically proves that you can make, that the transition is important, right? Because what you basically do here is you go more and more through the transition, and since we have started in the calculation, we have used parameters where we are just at the upper end of the transition. We start basically here in a case where it already increases with small excitations, right? But in principle, it should be. I come to that a little bit later. A sigmoidal curve, right? And that's what Matthias would call an all or nothing transition. It's not really all or nothing. It's a sigmoidal change. You can
terminology.
Huh? You have used it, right?
Yeah.
Yeah, but it is. You can go halfway, for example, right? That all or nothing implies. I show you an all or nothing pulse and later in the presentation. So now you can, since you go through the transition. Did I switch something off? stopped working, right? Sounds does it work again? Yeah, it works again. You go through the transition, and you can, for each density, and each density is associated to an enthalpy also. You can calculate the enthalpy of a part. The enthalpy is basically the heat uptake of a membrane when you make it liquid. But if you go in a pulse from liquid to solid, there's heat coming out of it. And that is basically the latent heat of the transition, and that comes out. And you can calculate that. You can calculate a heat release rate, and I don't know. Yes, on off again. So how did you switch it on? I can keep it on my hand. I don't know. Do I have to press a button?
The on, there's an on button, there's one on button.
Okay, good. And now I should be back. So if you integrate that, you get a reversible heat release like this. Heat is released when you go into the solid region, and it's reabsorbed when you go liquid again, back and forth. And they compare these two curves, which are calculated from the model, with these curves, which are from the experiment, from a very beautiful paper by Ritchie and Keynes from 1985, where they have both probably shown the most exact and the most beautiful data that are known about this. So in principle you can reproduce the thickness change. Maybe it's out of in principle you can reproduce a reversible heat, also in magnitude, and you can I have a strong breath. Okay, now I come to the second part of the talk, which is about anesthetics. Since in the soliton model for the nerve pulse, the transition play an important role, everything that manipulates this transition will have an effect on pulse propagation and pulse excitation. So here we have a lot of different molecules that all cause anesthesia. These here are local anesthetics, procaine, lidocaine, bupivacaine, and so on. Articaine is the one that you get at the dentist. Yes, and the other ones here are more general anesthetics, gases, most of them, or many of them. Remarkably, xenon is an anesthetic. Okay? I believe that the fact that xenon is an anesthetic falsifies every single molecule-related anesthesia theory, and I will show you why. There is this very old finding from Overton and Bayer, from the turn of the century, 18th to 19th to 20th century. They have put anesthetics in a mixture of olive oil and water, and they measured how much of the anesthetic is in the olive oil, how much is in water, and they called this ratio a partition coefficient. Basically, how soluble is the stuff in olive oil. And if you plot this partition coefficient versus the critical dose that you need to anesthetize somebody, then you get, so that means 50% of the individuals are asleep. In those days they used tadpoles, they had an aquarium, and the tadpoles were swimming around, you added an anesthetic, and they counted how much anesthetics do I have to add before 50% of the tadpoles sink to the ground. Basically a measure for immobility. Right? And they found a straight line. In a double log plot it has a slope of -1, that means it's in exact inverse correlation, and this is so remarkable that Overton already speculated about this. This must have a physical explanation because it's absolutely unspecific. There's xenon in that, laughing gas, but even over several orders of magnitude you see the same correlation. All these molecules do the same thing. And you can interpret this plot as that at critical dose, the concentration of an anesthetic in a membrane which has oil-like features is always the same. So that means the concentration of xenon or the concentration of halocene or so at critical dose is exactly the same number. So that means it only depends on the concentration, but not on the chemical nature of that stuff. For that reason, anesthetic effects are also additive. Yeah, so you can take 50% of one anesthetic and 50% of another anesthetic, you get full anesthesia. So what has also been found is that if you add anesthetics to membranes, that you get a decrease of the melting points. So membranes melt at lower temperatures when you add anesthetics. That is true for all anesthetics we have investigated. This is just one of them, octanol, a long-chain alcohol, but propofol and lidocaine and all these local anesthetics do all the same thing. They lower that, and you can describe the lowering of the melting temperature with a very simple law, which is the freezing point depression law that originates from Van 't Hoff from 1886, I believe, and was I think also at the basis of his Nobel Prize that he won later. Very simple law that anesthetics are basically antifreeze agents for membranes. They lower the melting point and make the membrane more liquid. Now, when you plot the temperature dependence of the melting, the concentration dependence of the melting temperature versus the critical dose, you get exactly the same straight line for all the anesthetics, and that gives some kind of physical meaning to this Meyer-Overton correlation. The Meyer-Overton correlation is only a correlation and doesn't give any kind of explanation for what happens. But this here is deeper because it gives you a real physical phenomenon that changes when you add anesthetics, and it plays an important role in this soliton approach, or the electromechanical approach. So here I've plotted again the energy density versus the enthalpy. So this is the position in the nerve, that's the nerve, the soliton here. And I plotted the enthalpy and the energy density. And if you look at the scale, the enthalpy density is nearly 100 times larger than the energy density. That is because when you really distinguish between analytical mechanics and thermodynamics, you actually you use different the term energy has a different meaning. Right? If you compress a spring, in analytical mechanics, you mean mechanical work. You don't take into account that entropies might change or might be involved in that. And actually what corresponds to an energy in thermodynamics, to the potential energy basically of something, is the free energy. There is a paper by Helmholtz already who pointed that out, that the free energy in thermodynamics is equivalent of the mechanical potential. So if you plot the free energy instead of the enthalpy versus and compare that to the energy density, you get basically the same scheme. So what I mean is that the energy, when we talk about a soliton and we attribute an energy content to it, what we actually mean is a free energy change. It takes into account that we have molecules present that also change the internal degrees of freedom and have an entropy change in there. And the free energy change in a transition is basically zero. Right? So that means the closer you are to a transition, the smaller the energy density will be, and the further away you are, the larger it will be. That gives that introduces a temperature dependence into that concept. So now let's look at that. The chemical potential, that basically is the free energy per mole, is the enthalpy, which we have shown on the previous slide, minus T delta S, which is significant, close to transitions of lipid membranes. And you can also attribute a free energy density to that if you multiply the chemical potential difference with the amount of fluid phase that you actually have present. You can calculate from this profile the heat capacity, and the total energy of the pulse is an integral over the of the free energy density over the whole pulse. One can plot that. This is the heat capacity. This here would be the chemical potential difference. It's just zero in the transition. Yeah, it's positive below the transition, negative above the transition. And you can now also plot. The density change versus temperature, or you can now plot the density change versus the chemical potential difference, or the free energy difference, and that gives you this sigmoidal profile. Right? So if you are here and you add free energy to the system, that's what how I interpret what a stimulation is. A stimulation is adding some free energy to the system that has the potential to change the state of the lipid system. And it doesn't matter what nature that is. It could be electrical, could be mechanical, could be chemical, whatever you want. If you add more and more free energy to the system, eventually you move the system through its transition, yeah, and you get this sigmoidal profile. And that is what I call the stimulation profile. Yeah, so that means the stronger you excite something, the more response you get. And a profile like that would be called a stimulus response profile. It's something that is regularly tested in medical practice, and I'll show you that in a moment. But what we would expect if we have anesthetics in a memory, this is a calculated transition profile of a memory. We add anesthetic to that, transitions go lower and lower and lower, and the stimulus response profiles that we calculate from that look like this. That means they are more and more shifted to larger stimuli. That basically means if you add anesthetics to a membrane, it's more difficult to stimulate a pulse. That's the only thing that changes. You do not block the pulse. People in biology always claim that nerve pulses are blocked by anesthetics. I don't believe that this is true. Yeah, and we have evidence for that. So here, first of all, are two experiments done in our lab. And this is a lobster nerve. We have measured it in the absence and the presence of anesthetic. You see a shift here. What we added is lidocaine in this particular case. So we get a shifted stimulus response profile. This is the earthworm that really shows an all or nothing thing for reasons that I don't quite completely understand. But it is either on or off. Yeah, but that worm nerve is the only nerve where I have really seen that. Yeah, with lobster nerves it's not quite as clear. I think they are continuous. Yeah, but that is maybe open for debate. Now, there was a collaboration that we had, or collaboration is the wrong word, because we parted from each other in a fight. But we tried at least to collaborate. And this concept of shifting the stimulation threshold for a nerve pulse was tested in a clinical experiment on a number of real people. Yeah? So what people wanted to anesthetize is the median nerve, that is this nerve going from the shoulder to the hand. The nerve was stimulated somewhere here with electrodes. It was anesthetized with syringes going close to the nerve. Yeah. So there are 5 different injection points where 5 milliliter of anesthetics were added. It's quite a dramatic amount of stuff that has been added. It was all done with ultrasound imaging. Yeah, so you could see the needle, you could see the median nerve, you went close to the nerve and added an anesthetic and anesthetized that nerve. And then here in the thumb, which is where one of the nerve branches of the median nerve goes, there is a response, and that was measured with these 2 electrodes here. Yeah, so it was stimulated here, the response was measured here, and what comes, then you see an experiment like that. That is a student of mine, a former student of mine, Lars Musco, and some of my students actually volunteered to be part of that experiment. And so basically what you see here, this experiment failed. Yeah, it was incorrectly done. That's because you see that because the complete hand is moving. If you do it correctly, the hand should not be moving, but it's very nice for demonstration because you see that there's a stimulation, the hand is doing something. Yeah? But if you do it correctly, actually the hand doesn't move, you just see a little reflex in the thumb. That's the fact that the whole hand is moving is a mistake because there's another nerve in there, the ulnaris nerve, which you also have to anesthetize, otherwise you get the movement of the complete hand. Anyway, so what has been measured in this experiment is the following. The hand was anesthetized, and one could apply a stimulus up to 50 milliampere. Yeah, the unit for the stimulation, magnitude of the stimulation is amperes. They measure the current that they use for the stimulation. There is a threshold in there, this is this dotted line, the maximum stimulation, which is basically only an ethical concern. Yeah, so one is not allowed to sacrifice the student after the experiment, and therefore there is a maximum limit for how much current you can apply. And in the beginning, one didn't see the pulse because it was right of the maximum stimulation that we could give. But then we waited, yeah, and we waited a complete day, basically, and slowly anesthetic concentration in the close to the nerve decreased because it was washed out with time, and then these curves reappeared. This was after 2 hours, after 4 hours, after 5 hours, after 6 hours, and after more than 6 hours it was nearly back to normal. Yeah? But it was only completely back to normal if you measured after 24 hours. But what that shows here is just different concentration of anesthetics. You could also see after 1 hour profiles where you could only see half of the profile. That's why I'm very confident that the nerve was never blocked, because you can go half up the stimulation, and then you don't see the other part of that because of the safety regulations and the ethical concerns that one has. Okay, and this here on the left-hand side is again that kind of simulation that predicts two things. The first thing is, independent on how much anesthetics you get, you always get the same maximum response. Yeah? This should not be the case in classical models like Hodgkin-Huxley, because in Hodgkin-Huxley model people believe that you block sodium channels, and that should in the end decrease the maximum amplitude. The second thing is that you can shift the stimulus response curve by a factor of more than 10 if you wish, which is also impossible in the Hodgkin-Huxley model, because the stimulation threshold in the Hodgkin-Huxley model is something like 25 millivolts. The maximum amplitude is 100 millivolts. You can never have a stimulation threshold that's larger than the maximum amplitude. That doesn't make sense. So this kind of experiment is completely unexplainable by the Hodgkin-Huxley model, but it is consistent with the experiment. There is no stimulation limit. You can get basically shift the profile by as much as you want. And now every variable, so what we have done here is basically to shift the transition. Everything that shifts the transition would have a similar effect. So for example, applying pressure would shift it back to the left side, because that it moves transitions to higher temperatures, and that has also been observed that you can wake up anesthetized creatures. So that was my last slide. So there is this soliton picture. We have had a lot of fights about the term soliton. I think it's not really important. This kind of thermodynamic model for describing that has a lot of advantages. It unifies many different things that one can observe. First of all, it makes use of the transitions, and the transitions are basically the sensor for the thermodynamic variables. So that whatever you change has an effect on these transitions. That changes the nerve pulse. You get the possibility of soliton propagation because of the non-linearity of the elastic constant and the dispersion. You calculate the right thickness change for the soliton. You calculate the right magnitude and the direction, the sign of the heat release from it. You can explain, I think really explain what anesthetics are doing in that thing. And you can understand why pressure is reversing anesthesia. Yes, and I stop there. Thank you very much. Yeah, Klaus, later.
I have a question for the stimulation of arm experiment. The nerve is normally stimulated with a current of about 10 microamps if you're sitting close to the nerve, and you have used 50 milliamps.
Yeah, but you're not sitting close to the nerve. The nerve is in the center of the arm, and you are at the surface.
The question comes now. That was already stated. The question is, it has this high current, or such high currents to stimulate. That means that you have deviated the current quite nicely around. So how much is the effect of having the drug into the tissue and the swelling of the tissue, whatever you put into the tissue, on the effect to stimulate the nerve. So in other words, the current path might change over time. And so how much are you really determining the effect on the nerve, or are you basically determining the effect by the force on the current path to the nerve to stimulate? Can you distinguish between the two? See my question?
Yeah, approximately. I'm not sure I can answer that. Of course, what we see in the end is a clear effect on the nerve because we measure the response in the sum. Yeah, but
no, we can directly stimulate the muscle too. So it not necessarily happens in
No, but that's too far away. That's too far away.
That I think one can exclude it. But how I understood your question is rather, does the anesthetic have any effect on the tissue surrounding the nerve, and that the change in the stimulation amplitude is an indirect effect and not a direct effect on the nerve?
It's an effect by changing the current path.
Yeah. I don't know. I know this method here, this experiment, is a standard procedure that people use for people who have defective nerves. Yeah, so that means there's a lot of experience with doing experiments of that kind, but administering so large amount of anesthetics close to that was probably something new. I cannot exclude that there are different other effects.
I only see that the current densities or the current amplitudes are very high to what you expect if you're getting close to a nerve. And if you're close to a nerve, your current amplitudes are normally not more than a couple of 100 microamps.
Yeah, that's possible, but we cannot do that. We cannot open a student. We have to apply a stimulus from the outside.
Jean-Pierre Chacheux.
Thank you very much for this very interesting presentation. I am concerned by the relationship of the observable, what you see, what you measure, and the mechanism of the propagation of the nerve impulse. And I have no doubts about what you said and about the measurements that you carried on. I'm pretty sure that this is indeed what's happening. Now, is it the cause or the consequence of the propagation of the nerve impulse? There, I'm asking a question there. Because I was struck by the data on local anesthetics. If they were general anesthetics, this would be even more terrible.
We have done one experiment with general
You have a concentration which goes from 15 millimolar up to 85, which is a 0.1 molar concentration. It's an enormous concentration.
Yeah, it's just a model calculation.
Well, that's, I don't know, it's a calculation, but if you use such a high concentration of compound, then this raises an issue that indeed you may need to have this high concentration of whatever, of general anesthetic or local anesthetic to reach the target where they are actually acting. And this is just to open the discussion. Either you change the overall state of the lipid, which is generally accepted.
Well, generally accepted in a certain part of the scientific community.
You act on targets which are in relation with the lipid phase, but which are proteins, and which are acting actually as the main molecular mechanism of the actual change of ionic concentration which are taking place.
I don't believe that the proteins are the target for several reasons.
You don't know. You don't feel it out.
No.
So wait until I show my data. Because we have been spending a lot of time to identify the sites for, and I'm not the only one. There are many groups who have identified the site for general anesthetics as allosteric modulators of ion channel, or in particular like-conjugated ion channel, and this is supported by the fact that if you mutate these sites, then you alter the effect of the general anesthetic. So there is a relationship of causality there at this level. This is just to open the discussion.
Yeah, but if you have a molecular target,
Let
then
me
you
say, I
would
have no doubt about what you said, but I'm just asking whether this is the mechanics or not.
Yeah. Thomas, of course, you can answer, but we'll also talk about it in the round table. But feel free.
I have a question.
No, the question
going
was respond.
possibly.
Should I respond?
Wait a moment. You can respond, but we also will talk about this in the round table.
If you have a molecular target, that basically means you can separate the effect. If you have high enough concentration,
Yes.
you can completely block everything. And that's not what we have found in the experiment. We couldn't block it completely. We just needed higher stimulus for the measurement.
If you use tetrodotoxin, you know, this is a compound from the pufferfish. It's acting at 10 to the minus 10 molar.
But that's
And it's not
anesthetic.
at 0.1 molar.
Yeah.
And then the stoichiometry, the relationship of the stoichiometry of the compound and the lipid, which is present in the membrane, is extremely powerful. Here you have something which is consistent with So, nevertheless, the prototoxin is acting on the nerve from the lobster. You know that?
Yes.
Okay. So how do you explain that?
Well, that would have to have an indirect effect on the thermodynamics of the membrane. So I consider proteins as being things that have chemical potentials and add to the thermodynamics of the system.
Okay, maybe one more question.
It's more or less the same question. When you are going this question with pharmacology, for example, with TTX or other drugs, which are working in two things, working at a very low concentration, and second, the partition coefficient tells us that they don't enter the membrane. So TTX or in this case,
I will try TTX. TTX does not really change the membrane transition in a model membrane. So it doesn't really interact much with this one. I accept that point, right? This model is a purely hydrodynamic model and doesn't contain any mechanism for binding of poisons. And that has to become inside by...
I think the global, I agree, as I said start, I agree with the general picture. I say that I agree with the general overview of what you said, but this is not for me as a molecular biologist.
And it's definitely not complete. That's a simple model that does not really reflect the influence of poison status.
Okay, let's
I accept that.
one more. Are you capable of keeping short? Let's try this.
Me? Yeah.
I want to make a comment from the other side. We all make mistakes, we all make errors, and when we go to detail, there's almost nothing we can say where such things don't open. So we have enough time to talk about, and the wisdom is not to lose out of sight the big question. So I only want not to open the question, I want to note mistakes from my view. So I made a mistake, and Daniel is around who discovered it, and Matthias also, even very important, about the capacitance thing. I don't enter further into it, and a mistake on numbers, whatever. You made a mistake on the term of soliton, in terms that one has, as Heimburg-Jackson says, the soliton you talk about is not the soliton as we are used to it. I don't go into the detail. It's all a big question, which should not take us away from the big point. What is it? Jean-Pierre made a mistake after a great breakthrough with allosteric transition, and it was the first system which Manfred Eigen and so on. A long discussion, and actually the mechanism which I have proposed is allosteric, no question. Is allosteric, but to put it into a protein solely is a mistake in the historical, but I don't want to open the discussion. We have time for that. And for everyone, one can open this discussion, and I only want to point out, we have the competence here to have this detailed discussion. We should not leave out of sight the big question. What is the nervous impulse? How does it relate to the conscious processes?
I think the most important thing there is that the pulse has been measured to be adiabatic, and that is really something one should take very, very seriously in my opinion.
Okay.
Thank you very much. Thank you for keeping short.